So welcome. I'm going to be doing a layman's discussion of cleavage for viruses, and it'll be a review on furin-mediated protein processing in infectious diseases and cancer. So here's the outline. The review is going to be of the review paper Furin-Mediated Protein Processing in Infectious Diseases and Cancer, and this is in Clinical Translational Immunology. And I will have the reference down in the description below. So I'm going to start with this breakdown, and then I'm going to relate this to the SARS-CoV-2 pandemic that is going on right now. And it is important because SARS-CoV-2 has a furin cleavable sequence on the spike protein. And then we are going to be doing a discussion on if this strategy indicates that there's going to be a higher propensity for this SARS-CoV-2 to be able to cross into different species. So this is the paper. And abstract, the proteolytic cleavage regulates numerous processes in health and disease. So proteolytic cleavage just means that a protein is being broken down into two or more pieces. So one key player is the ubiquitously expressed serine protease furin. So ubiquitously expressed just means that this particular enzyme exists in many cells throughout the entire body. So ubiquitous means everywhere. So since it's expressed throughout the entire body, anything that utilizes this enzyme can find this enzyme throughout the entire body. So, and it is a serine protease furin, and the best way to describe this is that serine, which is this amino acid that I have right here, is within the protein structure of furin, and this serine alcohol group is able to be activated to do the attack onto the proteins. And that's all you really need to know about that. So mammalian substrates of furin include cytokines, hormones, growth factors, and receptors. Oh, sorry, I skipped. And it cleaves a plethora of proteins at polybasic recognition motifs. So polybasic recognition motif. So polybasic just means that there are multiple basic amino acids at that particular location. And so here, as you can see, I have one amino acid, and this amino acid is serine, and this is the alcohol group that is on the serine. But there are different amino acids that have different chemicals right here in this group. So the basic amino acids are lysine, arginine, and sometimes histidine. So instead of having this OH group, you will have some basic group that is over here. And so when someone says that there is a polybasic cleavage motif, there's a group on the protein itself that has these multiple bases on it, and then the furin and other enzymes like furin are able to recognize this motif and then cleave the target or the substrate protein into multiple pieces. So continuing, thus it is not surprising that aberrant furin activity is associated with a variety of disorders, including cancer. Furthermore, the enzymatic activity of furin is exploited by numerous viral and bacterial pathogens. So viruses and bacteria, they're small, they have small genomes, and it takes a lot of resources for them to make everything that they need to make. So rather than have all that information within their own DNA, very frequently they will utilize the mechanisms and the tools that the host body has to fulfill their own biological needs. So rather than being self-sufficient, they decide to hijack the processes that you or an animal just has going on within its body already. So, and thereby, it's enhancing their virulence and spread. In this review, we describe a physiological and pathophysiological substrates of furin and discuss how dysregulation of a simple proteolytic cleavage event may promote infectious diseases and cancers. So right here, it's saying that breaking up these proteins is an important signaling event. Your body and the body of many living organisms are highly regulated. So every event that happens, if it becomes dysregulated, then you might have unintended consequences of that particular action. So what they're saying here is since furin is throughout the entire body and it plays such an important role in making compounds activated or deactivated, that if this becomes dysregulated, you can have diseases such as cancer show up. So one major focus is the role of furin in viral glycoprotein maturation and pathogenicity. So a glycoprotein is just a protein that has a little bit of a carbohydrate on top of it. So glycoproteins, your body makes proteins, and these proteins are the machines of the body for the most part. And sometimes these proteins will have little bits of sugar that is put on top of them. And there are multiple reasons for doing this. One of them is to help tell the cell of your body that this protein needs to be released out into the wild or out into the open area. And sometimes it's used to signal to your body that this protein is your own. So you, as a living creature, you are going to put certain carbohydrates on your proteins, and that can tell the rest of your body that you made this protein and it's not something that they should attack. And think of this, I like to think of this as the blood types on red blood cells. So you can have different blood types. You can have A, B, AB, and O. And the only difference, or the main difference between those, is you have the glycoproteins that are on the surfaces of your red blood cells. And if you have AB blood type, your glycoproteins on the surface of your cells are going to have both A's and B's on them. And so your immune system knows that it shouldn't attack A's or B's. So when you get a blood transfusion with someone from the A blood type, your immune system, or the person with the A blood type, their immune system is not going to attack that because it recognizes that A. However, people with O blood type have neither. And so if they get a blood transfusion from someone with blood A type, then the immune system has not seen this A type. And so it'll think it's foreign, which it is, and then start attacking it. And this is why some people can die if they get a blood transfusion from the wrong blood type. So continuing on, one major focus of the role in furin of viral glycoprotein maturation and pathogenicity. We also outline cellular mechanisms recognizing the expression and activation of furin, and summarize current approaches that target this protease for therapeutic intervention. So this review paper goes into a lot of detail, but I'm going to be focusing it mostly on its review of furin and its review on the different viruses that it can work on to increase the pathogenicity of those particular viruses. So, introduction. The human genome encodes more than 550 proteases. So your own DNA will make over 550 different proteases or different enzymes that break proteins apart. These molecular scissors play important roles in essentially all physiological processes. They digest the proteins in our food, degrade misfolded or unwanted proteins, and regulate the trafficking and activity of numerous cellular factors. Proteolytic cleavage is certainly one of the most important post-translational modifications, generating a plethora of bioactive proteins and peptides with key roles in cell proliferation, immunity, and inflammation. So we're going to come back to this. So what is post-translational modification? So this right here, I've made this very rough diagram of what happens inside your cells. So you have your DNA, which is your genetic code. And then this is within the nucleus of your cell. And so DNA ends up getting transcribed into RNA. So you make a copy of your DNA as RNA, and this gets taken out of the nucleus. So this process is called transcription. And then after you have DNA transcribed into RNA, then different proteins will come along, or different enzymes will come along and translate it from RNA into proteins. So you have your instructions, you copy your instructions, and then you execute those instructions to make protein. And that is called translation in the green. So after you have your proteins that you have synthesized from amino acids, then you can modify those proteins in a post-translational modification event. So one of those events is adding sugars onto a protein. That's an example to make a glycoprotein. Another one is proteolytic cleavage, so breaking a protein into multiple parts. So this is an important role of proteolytic cleavage within the body. It's one of the most important post-translational modifications that you can do for creating the active components of an original protein. And the difference between a protein and a peptide is proteins are big chains of amino acids and peptides are smaller, relatively smaller chains of amino acids. So proteolytic cleavage is certainly one of the most important post-translational modifications, generating a plethora of bioactive proteins and peptides with key roles in cell proliferation, immunity, and inflammation. Not surprisingly, mutations in proteases and/or aberrant protease activity are associated with numerous pathological processes, including cancer, cardiovascular disease, and autoimmune disease. Intriguingly, also, many viral pathogens exploit cellular proteases for the proteolytic processing and maturation of their own proteins. Similarly, activation of bacterial toxins frequently requires cleavage by proteases of the infected or intoxicated hosts. So as I said earlier, some viruses and bacteria don't want to make their own proteases. Sometimes it's just better to use the proteases that exist in their host organisms. So for diseases of the human, that's what a lot of viruses do. A lot of viruses will use the proteases within your own body. They're not always going to be furin, but you have over 550 different proteases. So viruses and pathogens only need to use a few of these proteases to become activated, if that makes sense. So in recent years, modulation of protease activity has therefore emerged as a potential therapeutic approach in a variety of infectious and non-infectious diseases. One particularly promising target for therapeutic intervention is the cellular protease furin. This protease most likely cleaves and activates more than 150 mammalian, viral, and bacterial substrates. So this one protein that you produce, we know of at least 150 substrates that it can attack well, that it can split up well. And that's just one of the 550 proteases that you produce. So this protease most likely cleaves and activates, among them are viral envelope glycoproteins and bacterial toxins, as well as cellular factors that promote tumor development and growth if they are hyperactivated. Just so you know, an envelope glycoprotein is a protein that viruses use to generate the envelope that goes around the viral material of the virus. So if you look at a virus and it is an enveloped virus, that is going to be the outside of the virus. So what else? In this review, we summarize our current knowledge of furin-mediated protein processing in health and disease with a focus on the role of furin in viral protein processing. Furthermore, we describe extracellular mechanisms regulating furin activity at the transcriptional and post-transcriptional level. Finally, we present approaches that aim at modulating furin activity for therapeutic purposes and discuss their suitability for clinical application. So, if you ever see transcriptional and post-transcriptional, just know that transcriptional means that if it's going from DNA into RNA, that's the transcriptional level, and then post-transcriptional just means going from RNA to protein or going from protein to a modified protein. Transcriptional and post-transcriptional. That is all going to be covered by activities of proteases such as furin. So the family of proprotein convertases. So just so you know, a proprotein is a protein that can be split by an enzyme into its final form. So if you have your protein, it'll just be called a proprotein if it needs to be broken apart into its final proteins at the very end. So just some vocabulary. So furin is a member of evolutionary ancient family of proprotein convertases. Excuse me. Their similarity with bacterial subtilicin and yeast kexin proteases has coined the abbreviation PCSK. Proprotein convertase subtilicin kexin types. So biochemists love to use a lot of acronyms, but let me describe this to you. So a lot of proteins can have very similar structures to each other's. And therefore, to help organize all of these different proteins, we like to cluster them into families. And those families are oftentimes named by some of the earlier discovered members of that enzyme. So when it comes to PCSKs, we were able to discover subtilicin, bacterial subtilicin, and yeast kexin first. And we realized, okay, these proteases are similar to each other. And then when we started discovering new proteins that looked like these, we had to say, okay, these are all a part of the same family. And since we discovered subtilicin and kexin first, we will call them the subtilicin and kexin family. So now that's why they're called PCSKs, the proprotein convertase subtilicin kexin type. And humans encode for 9 members of this protease family. So PCSKs 1 through 9. And PCSK3 is representing furin. So right here in the graph, we see PCSK3, and an alternative name for this is furin, and these are some other random names. And here we can see the target sequence of this protease. So you don't need to remember this all too closely. Just know that it is a polybasic amino acid sequence that is used for signaling to furin that this is my substrate. This is the thing that I need to attack, and then it finds this, and then it'll cleave right where this arrow is at. So you see this sequence, and then it will split the protein into 2 at this spot. So continuing, PCSKs are well known for their ability to activate other cellular proteins. The proteolytic conversion of inactive precursor proteins into bioactive molecules has already been described in the 1960s. However, it took more than 20 years until furin was identified as the first mammalian proprotein convertase. So we knew for quite some time that these enzymes that break up proteins, we knew that that was an important part in making the active versions of biomolecules. However, it took over 20 years after learning this to find the first one in mammals. And the first one in mammals was furin that we discovered. Continuing, to date, more than 200 cellular substrates of PCSKs have been described, including hormones, receptors, growth factors, and adhesion molecules. So out of these 9 PCSKs that your body produces, there are over 200 cellular substrates, or 200 things within the cell that can react to, or that can be reacted by one of these PCSK molecules. So, and I'm just going to have the target sequences listed here for you to look at. It doesn't matter too much. Just know that these individual letters are different amino acids. So K's stand for lysine, R's stand for arginine, and then X's could be any other amino acid. And if they have an N here, then it's N number of unknown amino acids. But in the case of furin, it needs to be an arginine, and then you have some other amino acid, and then you could have an arginine or a lysine, and then another arginine. and then you'll split right after that. And that is the typical sequence, but a lot of these enzymes aren't exactly precise in what they do, but this is the stereotypical cleavage site. So continuing, although PCSKs are frequently co-expressed in the same cell and may cleave the same substrates, there is no complete redundancy, and the inactivation of individual PCSKs result in specific knockout phenotypes in mice. So there are a lot of different PCSKs within the same cell, and a lot of them can attack the proteins for each other. But as you saw in the previous graph, those sequences aren't exactly alike, and so they might not see the same sequences, or they might not see the other sequences as efficiently. And just remember that everything within the cell has become highly regulated throughout, or it needs to be highly regulated, because these are important proteolytic events. And so if any one of those proteins doesn't work the way it should, you can see the result in the phenotype, or you can see the result in the physiology of the creature if you knock out or remove or change the activity of one of these PCSKs. And even if the others are working just fine, you remove one, you change the way that the creature behaves. So, continuing on, so PCSKs 1 through 7 cleave their substrates after basic residues, with the typical recognition motif of lysine or arginine, then XN, and then lysine or arginine again. Doesn't matter too much. In contrast, PCSK8 cleaves after non-basic residues and is best known for its regulation of cholesterol and lipid metabolism by activating sterile regulatory element binding proteins, or SREBPs, transcription factors. So this might be a little difficult to explain, but just know that a transcription factor is another protein. And this protein, see how we go between DNA and that gets transcribed into RNA? Well, the transcription factors are proteins that bind to the DNA and tell the DNA, or tell the other proteins, how much it should be transcribed into RNA. So PCSK8s will cleave, so the PCSK8s will cleave the transcription factors and make them active. And then, if you have your DNA, the transcription factors will then go onto the DNA and then tell the DNA, I mean, that's not exactly what happens, but it will tell the cell whether or not it should transcribe the DNA into RNA. And this is a way that you can regulate the production of cholesterol, or low-density it also plays a key role in cholesterol metabolism, as it regulates low-density lipoprotein particle levels in the blood. So, however, this effect does not involve proteolytic cleavage of a specific substrate. It is mediated by a direct binding of PCSK9 to LDL receptors. With 2 FDA-approved inhibitors for treatment of hypercholesterolemia, so high cholesterolemia in the blood, so if you have a lot of cholesterol in the blood. PCSK9 is also the prime example of a protease that is successfully targeted for therapy. So when it comes to developing different treatments or different pharmaceuticals, a lot of times the pharmaceuticals will target specific proteins. And in the case for high cholesterol, we look to PCSK9 and PCSK8 as prime targets to develop treatments for. And by blocking their activity or altering their activity, we can then alter the transcriptional rates. And then after we alter the transcriptional rates, we then alter the amount of cholesterol that you have in your own blood. So it's a long process, and you could imagine that it's a messy process to come up with these treatments because it's so many levels above where the actual cholesterol or the actual therapeutic is happening, but nonetheless, it is effective in many cases. Or I shouldn't say many, in 2 cases with this, we have FDA-approved inhibitors. So moving on to furin, which is one of those 9 proteins, the role of furin in viral protein processing and pathogenicity, and I actually skipped a lot of the review to get down to here. Like bacterial and viral exotoxins, most viral envelope glycoproteins need to be proteolytically cleaved before they can mediate viral entry into their host cells. So in many cases, viruses exploit cellular trypsin or subtilisin-like endoproteases for this purpose. So the subtilisin-like endoproteases or proteases were the ones that I showed in the previous table, the subtilisin-like ones. However, there's another group of proteases known as the trypsin-like proteases. And just know that that is a completely other family of proteases that your own body produces. And not just you, but a lot of other mammals and a lot of other creatures will also make these trypsin-like proteases, as well as subtilisin-like proteases. And while subtilisin-like proteases such as furin require polybasic cleavage sites, so when you have multiple bases in a particular location, this is what furin requires. Trypsin-like proteases will also recognize monobasic motifs and cleave after single arginine or lysine residues. So the trypsin-like proteases can see your polybasic sites as well, because if you have 4 basic sites or 3 basic sites, you still have 1 as well. So a trypsin protease can see the polybasic site and cleave it, but they can also see the monobasic cleavage sites and then come to those locations and cleave those as well. And this process is also highly regulated even for the trypsin-like proteases. So notably, the dependency on specific proteases can also be an important determinant of tissue tropism and viral spread in an infected organism. And so tissue tropism, the best way to describe it is which cells in the body can host a virus. So some cells have receptors that allow a virus to come onto the cell, right? So the virus can come onto the cell. But if this particular cell doesn't have the protease available to allow the virus to go in, then the virus will just stick there and not infect the cell. So what we're saying is, depending on what proteases the cell produces, that is going to determine what cells are vulnerable to a virus coming in, or to a virus attaching and then going in. So if that event is specific to a specific cell, then only a few cells in the host are going to get infected. And but on the other hand, if that event is ubiquitous, then many cells can host the virus. So if you could imagine, if a virus only has a monobasic cleavage site, it comes to the cell, then it requires a specific protease to allow it to get into the cell. But if it has a polybasic site, then we know that furin and some other proteases of that same family are expressed throughout the entire body. They're expressed ubiquitously. And because of that, it doesn't matter. If it can attach, then it's going to get in, or there's a high likelihood of it getting in. So going back to this, for example, to give an example, avirulent or non-virulent, so a very benign Newcastle disease virus, NDV strains, harbor a monobasic cleavage site in their fusion protein. and the fusion protein is just the protein that causes the attachment. Excuse me. So the fusion protein, which we call it F because we love acronyms in biochemistry, fusion protein F, and result only in local infections, mainly in the respiratory tract, since expression of the receptive host proteases is limited to a few cell types. So, in contrast, the F protein of virulent NDF strains can be cleaved by furin or related pro-protein convertases that are ubiquitously expressed. Consequently, these viruses are able to spread systemically and cause high rates of mortality in infected birds. So, if you get a disease in you, or if you're a bird and you get an NDV virus strain, if the virus is only able to exist in a few cell types, let's say in the lungs, then that is where your immune system can focus its resources on, and the virus really can't spread easily in the rest of the body because the other cells in the body of the bird are not producing the enzyme that makes the virus go into the cell. However, the virulent or the dangerous viruses, you get in, or the bird will take in, gets into the lungs or whatever other way it can get in, and it multiplies there, but then it can spread easily because it has a polybasic cleavage site rather than a monobasic one. So it's not limited to one spot, it can go wherever it wants. And not only does it make the disease more dangerous, more widespread, but then it also would mean that it is easier to become infected because now the virus doesn't need to find the very specific cell to host it. It could just infect any cell that is able to host it. And frequently, luckily, your skin is dead, so it's not going to be able to do that. But a lot of it just needs to, if a virus is able to have many different routes of infecting you, then that virus is more infectious and it's easier for you to catch that virus. So continuing on, another well-described example is the cleavage of influenza A virus, hemagglutinin, or HA, because we love our acronyms. Hemagglutinin, it's the virus protein and it sticks to blood cells. So hema meaning blood, glutenin, glue-like. So it's a protein that likes to stick to blood cells for whatever reason. In contrast to low pathogenic avian influenza A viruses, their highly pathogenic counterparts harbor a polybasic furin cleavage site in the HA protein. So in this hemagglutinin protein that is on the surface of the virus, this is what helps it get or attach to whatever its target cell is. But the more dangerous ones have a polybasic furin cleavage site, which means it is easier for them to find the protease to allow them to go into the cell. So continuing, thus the ability of viruses to exploit furin may have a drastic effect on their pathogenicity. To date, furin-mediated cleavage has been described for envelope glycoproteins encoded by numerous evolutionarily diverse virus families, including herpes virus, coronavirus, flavivirus, togavirus, bornavirus, bunyavirus, filovirus, orthomyxovirus, and all these other viruses. So although viral furin substrates generally harbor the canonical polybasic cleavage site, timing and subcellular localization of furin-mediated activation may differ substantially between virus families. So stopping right there, sometimes, very frequently, the viruses aren't just using the proteases to get into the cell. Sometimes they are also using the proteases to produce the active versions of their proteins as they're being created. So really, viruses and other pathogens are using these proteases in in many different steps along their life cycle. So this is just one thing to consider. It's not just for getting in the cell, it's also for producing the cell in the first place, or producing the virus in the first place. So since furin and viral glycoproteins both enter the secretory pathway, proteolytic activation can occur at different steps of the viral replication cycle. While the envelope proteins of some viruses are cleaved in the producer cells, others are processed in the extracellular space or during entry into their target cells. And the following sections highlight the characteristics of a few selected viral glycoproteins, their processing by furin, and the importance of furin-mediated cleavage for infection and pathogenicity. So here we have a table of many different types of viruses, and this is obviously not a comprehensive list. There are an innumerable number of different viruses in each of these categories. But here they have an example of the species of virus, what protein, and these are all acronyms. You don't need to memorize them, but on these proteins, they will give you an example of the exemplary cleavage site, or this polybasic cleavage site that is within this particular protein. So let's go to coronavirus, for instance, the infectious bronchitis virus has this polybasic cleavage site where it's arginine, phenylalanine, arginine, and arginine again, and then clips right here. So you can look at this table if you want on the website. But yeah, that's all you really need to know, is that all these different viruses have these exemplary cleavage sites that they take advantage of. So here's a good example, or a good diagram to help you understand what are the different strategies that these viruses are utilizing by taking advantage of furin. So for instance, with HIV, here we see the HIV protein, or the HIV particle being produced. So here you have your cell, and here you have your nucleus. And what is happening is HIV gets in and it takes over your cell, and then your cell starts producing the proteins of HIV. And these are here. And as these proteins are being secreted out into the cell, they are going to be processed by furin into their active form. And so they get processed from the immature envelope protein into the mature envelope proteins by the furins. And then these mature ones end up going to the cell membrane. And then once they're on the cell membrane, they start coalescing around each other to make the envelope for the developing HIV virus. So that's one strategy. The other strategy, like with dengue, I don't know how to pronounce it, is the virus, rather than forming up here in the cell membrane, the virus pretty much already forms within the cell, but it ends up getting cleaved by furin. So these PRMs, that's just an acronym, get cleaved by the furin, and that gets it ready to be completely secreted out into the environment again. So that's when these are both protein processing during the virus maturation. Here we have protein processing during virus entry. So here, example with dengue virus again, you have your uncleaved PRMs, and these uncleaved ones end up binding to the receptors on your cell, and then it gets sucked into, it gets endocytosed, or the cell thinks that this is something that I can just take in. And then once it's in, the furin molecule is necessary for cleaving the PRMs and then allowing the genetic material that is right here to then become released into the cell. So that's one example with dengue. And then here we have another example with HPV, where the virus sticks to a receptor, and in this particular case, it's a proteoglycan, but it sticks to a receptor, and then furin comes later. And remember, furin is ubiquitous. It can actually go in between, outside and inside of the cells. It can come up and then cleave the proteins of the HPV casing, and then that allows it to get taken up into the virus. And so here, furin clips activates it, and then that allows it to become or to go inside of the cell. So now we're going to go into a little bit more detail of the different viruses. So 40 minutes. Okay. So retroviral or HIV, retroviral glycoprotein trimers, such as those of human immunodeficiency, Roux sarcoma, or murine leukemia viruses, are proteolytically processed and activated in the producer cells. In the case of HIV-1, the GP160 precursor of the viral envelope protein is cleaved into GP120 and membrane-anchored GP41. So, step back. What happens is the HIV genetic code, rather than make two different proteins, it has decided that it's better to just make one large protein and then split that protein into the two parts. One of those parts is the GP20, and the other one is the membrane-anchored GP41. So the GP41 stays in the cell membrane, while GP120 basically sticks to the GP41, and that is, we'll go into more detail later. Oh yeah, and just so you know, a non-covalent interaction is just when you have two molecules or proteins that are sticking together without a formal chemical bond in between them. So there are ways to have things stick together without necessarily having a chemical bond in between them. So that's just what non-covalent interactions mean. So cleavage occurs in intracellular compartments before the assembly. And it remains associated through non-covalent interactions. So cleavage occurs in intracellular compartments before the assembly of virions at the plasma membrane. Notably, proteolytic processing of ENV depends on correct N-linked glycosylation, as aberrant carbohydrate side chains may result in subcellular mistrafficking or sequestration of ENV. So, a lot of jargon right there. So, an N-linked glycosylation. So, remember when we were talking about glycoproteins, where you have your protein and then you have a little bit of a sugar that can get connected onto it. An N-linked glycosylation is when a carbohydrate is added to a nitrogen on the protein. So nitrogen N-linked. So carbohydrates are added to a nitrogen on a protein. This technique is used for secreting proteins outside of the cell, and this can be important to protect the proteins from the immune system. So this functions multiple roles. Glycosylation is important for telling the cell where to move the proteins that it glycosylates, especially if they're being secreted outside. A lot of times you need to have the sugars on them to protect them or to convince your body not to attack them. So in the case for HIV, it is one of the reasons why it is difficult to create antibodies for it is it's also a glycosylated protein. It's a secreted protein. So your own cell will add sugars onto the proteins, and since those are your own sugars, then it is more difficult for your own immune system to recognize them as foreign. So that's just one of the strategies that viruses can employ to circumvent your immune system. So continuing on, and they also say that if this process is screwed up, then the HIV virus might not be produced correctly because of the mistrafficking, or they're moving proteins into the wrong locations. So most likely, HIV-1 takes advantage of the redundancy of several pro-protein convertases, recognizing the polybasic cleavage motif in ENV. So the ones that they think are furin, PCSK5, PCSK6, and PCSK7, and they have all been shown to cleave this GP160 in cells, albeit with different efficiencies. So if you're a smart virus and you know that you have to break the GP160 into two constituent parts, you're going to want to do that using an enzyme that you can find in many different cells. Or maybe you can't find the particular enzyme you want, but what if there's another enzyme similar enough that is able to do the same thing? That is the case here with HIV, or the presumed case, is that it doesn't just use furin, it can use all the other proteases that can recognize this polybasic amino acid sequence. And this is important because if it required a much more specific protease, then it wouldn't be able to duplicate as easily. It wouldn't be able to create the new viruses as easily. So continuing, notably, in vitro, or in a test tube, or in glass, cleavage experiments using recombinant proteases did not always reflect cleavage efficiency in transfected cells, and the relative contributions of individual PCSKs to HIV-1 maturation in vivo remain unclear. So what they're saying is there's a difference between what you can see in a test tube versus what might be seen in an actual living organism. So it's hard to know what exactly is going on, but they said maybe this might be an effective strategy that HIV is employing. Interestingly, HIV-1 envelope harbors a second polybasic cleavage site, about 8 amino acid residues upstream of the major one. And although cleavage at this site does not result in fusiogenic ENV species, about 15% of all GP160 molecules are cleaved at this position, at least in case of the cell culture adapted HIV-1 clone LAI. So what they're saying is HIV doesn't just have the main polybasic cleavage site, they have another one that is not too far away. And they said that if it cleaves at the wrong spot exclusively, it doesn't make an HIV molecule or an HIV virus that is effective at entering the cell. But I assume this research was they had the two polybasic cleavage sites and they just completely removed the active one, and by removing the active one, they saw whether or not that virus was infectious still, because one would assume, oh yeah, if you can cleave here instead of here, maybe it's still active. And they did that experiment and they said, no, it doesn't seem like this is completely redundant, but it could be a different strategy. It could be that with the HIV molecule, maybe some of them are cleaved here and some of them are cleaved here, and by having different cleavage sites, it makes it harder. I mean, I'm just making a guess right now, it might be harder for your immune system to recognize what protein it needs to attack, because if it can get cleaved in different locations in the final virus, then you have a bunch of different shapes and it is much more difficult to create an effective antibody for that. And who knows if that's actually what's happening, but that is my guess. So in some cases, GP160 escapes intracellular cleavage and may be incorporated as an unprocessed precursor into budding virions. Whether these envelope molecules may be processed extracellularly by shed furin is unknown. In this context, it is noteworthy that membrane-bound plasmin has been shown to convert extracellular GP160 into GP120 and GP41. However, it remains to be determined whether plasmin-mediated cleavage results in fully infectious HIV-1 particles. So what we're saying is there's another protease, plasmin, that is in the extracellular space. It's not within the cells, it's outside of the cells. And let's say you create, let's say the HIV virus is made without being proteolytically cleaved by furin. Well, it's a useless HIV particle, but it might be possible you have this useless virus, and then it might be able to get cleaved while it's floating around in the body. And if that ends up happening, then it's possible, it's possible that this immature or uninfectable virus has been activated to become infectious. We don't know this. They decided not to continue with that experimentation for some reason. So let me continue. And yeah, okay. So continuing on, highly pathogenic avian influenza A viruses. So influenza A virus hemagglutinin, so the HA, can be cleaved and primed by a variety of cellular proteases. Even bacterial proteases may promote influenza virus spread by cleaving HAO and HA1 into HA1 and HA2. Both subunits remain linked via disulfide bond and form trimeric structures. So just, a disulfide bond. So the influenza virus makes the HA0 protein, and you have this HA0 protein, and then it gets cleaved by the proteases, by a large variety of proteases. But they still stay connected with a disulfide linkage. So a disulfide linkage is a strong covalent bond between sulfur groups on an amino acid. So this is what gives hair your rigidity. So if you get a perm, for instance, they destroy the disulfide linkages in the proteins of your hair, and then they change the shape, and then they allow those disulfide linkages to reform in the different shape. So this is what's sort of what happens with the hemagglutinin protein, is that it gets cleaved, but it gets cleaved in the amino acid sequence, but there's still this disulfide linkage in between the two parts of the hemagglutinin molecule, so it still technically is connected. So I don't know exactly how it activates it, just know that it probably changes the shape significantly to make the active form of hemagglutinin. So while HA1 binds to the sialic acid receptor on viral target cells, so the HA1 is the part that sticks out of the virus and helps bind it to the cell. HA2 harbors the fusion peptide that mediates fusion of the viral and cellular endosomal membranes. So HA1 helps bind the virus to the cell, but HA2 is the peptide that basically allows the virus to go into the cell. So HA cleavage can occur within producer cells upon release of virins from infected cells or directly prior to entry into new target cells. So what they're saying is this HA, as long as it gets cleaved at any point of its process, it is then infectious. So yeah, that's basically all I have to say about that. As a general rule, hemagglutinins of mammalian and low pathogenic avian influenza A viruses cannot be cleaved by furin, as they usually only harbor a mono or dibasic cleavage site. Instead, they depend on trypsin-like proteases such as transmembrane protease serine S1 member 2, TMPRSS2, because we love our acronyms, or human airway trypsin-like protease, HAT. Expression of such trypsin-like proteases is largely restricted to the respiratory and gastrointestinal tract. Just a quick side note, this TMPRSS2 is presumed to be, I shouldn't say presumed, this is the protease that activates the original SARS virus. And this is exclusively, I don't know how exclusive, but it is found in the lungs. There might be low levels of expressions in other areas, but in the lungs, that is where it's more highly expressed. So continuing, in contrast, H5 and H7 hemagglutinins of a large number of highly pathogenic avian influenza A viruses, HPIVs, can be cleaved by furin or PCSK5, which are present in many cell types. So what they're saying is, if you only have the mono or dibasic cleavage site, it requires either TMPRSS2 or HAT, and those are in exclusive areas such as the lungs or in the digestive tract. But if there is a influenza virus that has the polybasic cleavage site, then it can take advantage of either furin or PCSK5 within human, or I don't know if that's within humans exactly, or if they're talking about avians. I think we're talking about humans right now. Since it's present in many cell types, it can cleave in a lot of those other locations as well, and it's more pathogenic as a result. This is because they acquired a polybasic cleavage site upon insertion of additional lysine and/or arginine residues. Duplication of lysine and arginine residues in HA is facilitated by polymerase slippage, as the amino acids are encoded by purine-rich codons. So, a lot of jargon, polymerase slippage. So, when it comes to polymerase slippage, think of polymerase. So, DNA and RNA, those are polymers. So, polymerases are machines that can polymerize a new strand of DNA or RNA. So when copying the genetic code, sometimes the machinery can slip backwards or forwards. And this is a way that you can duplicate or remove amino acids from the protein that is a result of this particular event. So to do a recap, just so that we're all on the same page, you have your DNA that is in your cells, you have RNA, and the DNA gets transcribed into RNA, and then the RNA gets translated into protein, and then the protein can be post-translationally modified into a modified protein. So polymerases are things that can duplicate DNA or RNA, and if a polymerase slips, it can lengthen or shorten the DNA or RNA that it's trying to produce. And the children will be mutants, for lack of a better word. They'll be mutants, and if they might be a little bit better or worse at surviving than their parents. So in the process of duplicating a DNA or an RNA, so let's say you have a virus that uses RNA as the genetic material. If it tries to duplicate itself with whatever enzyme and the enzyme slips, it is possible that you can duplicate a section of the RNA. And if a section of the RNA gets duplicated, then the protein will have a little bit longer, a duplication of some amino acids that are on that. And so that is one strategy or one method by which you can go from a mono or dibasic cleavage site into a furin cleavage site or a polybasic cleavage site. And if that particular mutation allows a virus to multiply more rapidly, then through evolution, it is going to become the major, it's going to multiply faster than its competition. It'll outcompete the other viruses. And so it'll be a mutation that has caused higher survivability of that virus, of the mutant. So, and excuse me, the reason why you can have this slippage is that you have purine-rich codons. So DNA and RNA are made out of nucleotide building blocks, and the nucleotides can have either pyrimidine or purine parts. So some sequences of DNA and RNA are purine-rich, and in this situation, the machinery, the polymerases that duplicate the DNA and RNA, they are more likely to slip if the area is purine-rich. And so this is an example of DNA. So here you have your DNA, and you have your two strands, and these are the individual units or monomers of the DNA polymer. You have your phosphate backbone, you have your sugar, and here you have your nitrogenous base. And so these bases right here can pair up with each other specifically, And here we have guanine, and here we have adenine, and these are purines. This particular shape with this 6-membered ring and this 5-membered ring fused to it, this is a purine, and so is this one. And these are pyrimidines right here with the thymine and cytosine. Now, when you have purine-rich codons, it is just more easy. I don't know exactly how this works, but it is easier for the polymerase to slip back and forth in those areas. So let us continue. Where were we at? So duplication of lysine residues. Okay, so instead of the prototypical arginine X, lysine or arginine, and arginine motif, some HPIVs harbor a suboptimal or not as effective this site, and I shouldn't say but it's lysine blank, lysine or arginine, and then arginine. So this is suboptimal. It doesn't work as well, but it still works. Cleavage motif. So proteolytic cleavage at this site is only efficient if additional positively charged amino acids upstream of this cleavage motif are present, or if attachment sites for masking oligosaccharide chains are missing. So, talking about oligosaccharides again, or talking about glycosylation again. So sometimes, if you have your protein, and your protein has a polybasic site, sometimes the protein can have glycosylation on it. So it can be a glycoprotein. You can have the carbohydrates that are added onto it. And if the carbohydrates can block this site, it might make it more difficult for a protease to come in and attack it. And that can lower the virulence of a particular virus, or it can lower the ability for this protein to be catalytically cleaved. But on the flip side of that, if you have glycosylation nearby this cleavage site, it might be able to protect that cleavage site from your white blood cells from making antibodies for that particular site. So it's a double-edged sword right there. Continuing on. So notably, a subset of H9N2 lowly pathogenic avian influenza A virus strains also harbor the RSKRs, or the RSRR sites, that are not only cleaved by trypsin-like proteases, such as TMPRSS2 or HAT, but also by the PCSKs. However, this cleavage is only efficient in the presence of very high amounts of furin, or upon mutation of glycosylation sites in HA. So what they're saying is, with this particular strain, this strain has the polybasic furin cleavage site. It has the polybasic site that allows it to be cleaved by many different proteases. However, it is protected by glycosylation. So if you want to make it more pathogenic, you have to delete the glycosylation site somewhere on that protein, and then it exposes the polybasic furin cleavage site. But everything's a trade-off. Everything is evolutionarily based, right? So maybe if you remove it, it might not necessarily make the strain more pathogenic. But they're saying in this case it is. In this case, if you do that mutation and remove the glycosylation, the virus has become more pathogenic in this particular case. Thus, the ability to exploit furin for efficient HA cleavage and the associated increase in pathogenicity are not only determined by the presence of furin, consensus target site, but also by adjacent residues and the absence of masking oligosaccharide chains. And so an oligosaccharide is just a small carbohydrate. So oligo meaning many, and saccharide sugars. So having many sugars is an oligosaccharide. It's a small carbohydrate chain. And I just want to make a note here. I've read in another paper that for SARS-CoV-2, the furin site might have nearby glycosylation through through computational studies of the proteins on SARS coronavirus 2, there's an indication that there's some glycosylation that's happening very close to the polybasic cleavage site of SARS-CoV-2. However, I don't think it's fully known whether or not it is glycosylated. It was just a computer study. So, okay, getting on to the better diseases. So, Ebola and Marburg viruses. So, furin cleaves the glycoproteins, GPs, of Marburg virus, MARV, and all 5 Ebola virus species into a large N-terminal subunit, GP1, that mediates receptor binding, and a small membrane-anchored C-terminal part, GP2. So, stopping here, the GPs are split by furin into 2 different parts. And membrane-anchored means a part of the protein will stay within the cell membrane. And let's see, to know what an N-terminus and a C-terminus is, that's a little bit of jargon. So here, I've given an example of an amino acid sequence. So here is one amino acid, here is another amino acid, here's another amino acid, and another. As you can see, on this side, it ends with an amino group, or the NH2. It ends with an N. And this is called the N-terminus. And over here, on this side, it ends with the acid group. So they call it the C-terminus because it's the carbon acid. Technically, it ends with OHs or an O, but we call it the N or the C. So amino acids have amino groups and acid groups, and then they are fused into these amide groups, and they make the polymers. But you can't, as long as you have a straight chain of a polymer, you're going to have one end that still has a free amino group, and you'll have another end that has the free acid group. And so the side of the amino is called the N-terminus. The side of the acid group is called the C-terminus. So going back to this, furin cleaves the glycoproteins of into a large N-terminal subunit, GP1, that mediates receptor binding, and a small membrane-anchored C-terminal part, GP2. So they're saying you split the protein into the N-terminal part that helps it stick to the cells that it's trying to infect, and then the C-terminal part contains the fusion peptide, which forces it to go into the cell. So MARV and human pathogenic Ebola species harbor the canonical furin cleavage site RXK or RR cleave. In contrast, the GP of the closely related Reston virus, which causes asymptomatic infections in humans, is processed less efficiently by furin as it carries the suboptimal cleavage site KQKR. Surprisingly, however, uncleavable GP mutants of highly pathogenic Ebola viruses are able to mediate infection, and furin-mediated cleavage is not required for replication in cell culture. Furthermore, an EBOV mutant lacking the furin cleavage site replicated efficiently in non-human primates and showed no difference in disease progression or lethality compared to wild-type virus. Thus, the high conservation of the furin cleavage site among different Ebola virus species is surprising, and that it remains to be determined whether furin-mediated GP processing plays a role in the natural reservoir hosts of this virus. So, stepping back, we can see that with the closely related virus to Ebola, it has the polybasic cleavage site, but it is a far less effective one. So we're presuming, at least to a small degree, that since it has a suboptimal polybasic cleavage site, that it is less virulent when it gets into the human body or any body. But with Ebola, even when you remove the furin site, it's still pathogenic. And does this mean that the natural host doesn't even use this site? But if it doesn't, why is it conserved? Why do you find it all the time? when you look at Ebola species. So, I mean, that's just, this is something that science has yet to figure out, but they had to include that in there. Continuing on, notably, EBOV GP gene harbors an RNA editing site sequence and may not only express full-length GP, but also a soluble form of the glycoprotein pre-SGP that lacks the C-terminal transmembrane domain. So remember, the original GP has both the N-terminal GP1 and the C-terminal GP2 units, and these two stick together. However, this pre-SGP does not have the C-terminal transmembrane domain. So rather than sticking to the membrane, it ends up free-floating in around. So pre-SGP is missing the part that sticks to the membrane, but still has a cleavable site. And continuing on, intriguingly, pre-SGP harbors another furin recognition site and is cleaved into mature SGPs and a short so-called delta peptide. Both of them are ultimately released from infected cells, although pre-SGP is produced in higher amounts than GP. Its role in viral replication is under debate, although among others, SGP has been suggested to serve as a decoy antigen to act as a structural substitute for GP1 and to induce apoptosis of uninfected lymphocytes. So it is unknown exactly what it does. But here they suggest that maybe this SGP can replace some of the function in the virus. So remember, the normal GPs are making the outer part of the Ebola viruses. Is it possible that some of these SGPs can replace the GPs within it? Because if you, let's say you replace only some of them, maybe the virus is still infectious and it's still able to do what it wants to do. Another thing would be if it is making these SGPs and releasing them out into the body, then as the body's trying to make antibodies, it might make antibodies for the SGPs rather than the GPs. So it's making all these antibodies and they're not working. The antibodies are not working on the real virus. It is working on the decoy SGPs that are floating around in solution. We don't know exactly what it is, and we can't just inject people with Ebola to see what the actual cause is. But that is one of the presumed, or a few of the presumed rationale or strategies that have been ascribed to this SGP protein. Oh, and the other one is that it might be able to kill. So apoptosis is basically killing a cell. So if it can kill the lymphocytes or some white blood cells, then that might help the Ebola virus evade the immune system. So that's another strategy that they suggest. Moving on to flaviviruses, and this is important because flaviviruses, a lot of them are mosquito-borne illnesses. Flavivirus RNA is translated into a single large polyprotein. Polyprotein just means many proteins all in one. Polyprotein that is cleaved by cellular and viral proteases into all structural and non-structural proteins of the viruses. The structural proteins, flaviviruses make one giant protein, and then this giant protein is broken down into the individually functioning pieces. And this isn't necessarily by furin, but that is a strategy that it employs. The structural proteins comprise the envelope proteins PRM and E that are incorporated as the PRM-E heterodimers into budding virions. PRM acts as a chaperone and facilitates correct folding of the E glycoprotein. So a chaperone in biochemistry, chaperones are proteins that make sure that the other proteins are folded correctly. So you make your amino acid sequence when you do translation, and then after you make that sequence, it has to the amino acid sequence has to fold in on itself to make the final protein structure. And oftentimes, if it folds, it can fold incorrectly. So what chaperones do is chaperones can recognize when, well, okay, there are different types of chaperones. In this particular case, the PRM, let me make sure that I have the right ones. So PRM is acting as a chaperone for the E. So you make your protein, it splits into PRM and E. The PRM is actually making sure that the E portion gets folded correctly into its final active form. And so if this doesn't happen correctly, you could imagine that the E protein will not form the correct structure, at least a large amount of the time. So, many flaviviruses bud into the lumen of the ER and enter the secretory pathway. In the acidic milieu of the trans-Golgi network, or the TGN, a furin cleavage site is exposed, and the PRM can be cleaved into mature PR and M proteins. Thus, furin-mediated cleavage of viral glycoproteins occurs only after its incorporation into newly formed virions. This is in contrast to viruses such as HIV-1, whose envelope proteins are cleaved before assembly. The PR peptide remains associated with the E protein until the virion is released from the cell, therefore preventing premature, unintended fusions with the membrane of the producer cell. Furin-mediated PRM cleavage is essential for replication of flaviviruses such as tick-borne encephalitis or dengue fever. So, in some cases, PRM molecules escape furin processing in the producer cells and result in the release of immature or partially mature viral particles. Partially mature virions are still infectious since low amounts of mature M are sufficient to mediate fusion with target cell membranes. Furthermore, uncleaved PRM may participate in virion attachment to target cells and can be cleaved by furin during the entry process in the acidic milieu, I don't know how to pronounce it, of endosomes. The relative contribution of PRM processing during viral entry into new target cells, however, remains to be determined. So the ratio of mature to immature PRM depends on a variety of factors, including the producer cell type and the flavivirus species. For example, dengue viruses are known to release high amounts of immature or partially mature viruses, most likely because of a conserved acidic residue within the furin recognition site. Importantly, the content of the PRM in viral particles also affects antibody recognition and consequently antibody-dependent enhancement of dengue virus infections. Thus, furin-mediated protein processing may once again markedly affect the outcome of infection. So, one of the things that I would like to talk about is the virus, for the flaviviruses at least. The virus is made within the cell, but then it is processed into its final form later. And this helps prevent the newly formed virus from accidentally fusing with some of the parts inside of the cell. So proteolytic cleavage is a mechanism by which the viruses will only be released, or what I should say, will only fuse with the membrane after it's been released into the virus. So antibody-dependent enhancement as well. I'll do another video on this, but this is your antibodies can be used against yourself to make a disease worse. So sometimes if your body tries to make antibodies against a disease, sometimes the antibodies aren't good enough to do it. Or sometimes they can do it, but they can just barely do it. So you might be able to recover from the disease once. But later, and you might be able to do this because you produce a lot of antibodies at once. And so even if the antibodies don't work very effectively, if you create so many of them, then they might be able to defeat whatever virus they're trying to attack. Now, a worry comes when, if your antibody levels of this particular disease gets low enough and you get infected again, there's an event called antibody-dependent enhancement, where the antibodies you do have in your system, you don't have a lot, but they stick to the incoming virus. And then, since they stick to the virus, but not very effectively, and it doesn't coat the whole thing, you basically added a signal, your antibody signal, which ends up helping the virus stick to a white blood cell. And there is a mechanism using antibodies to have a virus get incorporated into a white blood cell without destroying the virus. And this is antibody-dependent enhancement. And by doing that, you can, if you get infected a second time, you can really give the virus a really large head start the second time around. So this is why when people catch dengue fever or dengue, they tell them not to catch it again because you might have hemorrhagic fever the second time around. And antibody-dependent enhancement is the driving mechanism by making the second infection a lot worse. So continuing on, so this is as a recap of the dengue virus. You make the virus, and then it gets moved, and then it gets cleaved by furin here, and then as it's, and remember, inside of here, it's acidic, and this is what helps prevent it from fusing with the wrong membrane. And then once it gets out here, it gets released, and then is free to, wreck crap up in other locations. I'm gonna take a quick break. Okay, we're almost done then. So papilloma viruses. While furin plays a key role in activating envelope glycoproteins of a variety of viruses, its activity is also exploited for the cleavage of other viral proteins. One example is the cleavage of the L2 protein of papilloma viruses. Together with the major capsid protein L1, this minor capsid protein builds the viral capsid. So L1 is the major capsid protein, and L2 is the minor one, and they make the protein structure of the virus, known as the capsid. The furin cleavage site is located close to the N-terminus, and remember the N-terminus is the nitrogen, or the amino acid, or the amino containing side of the amino acid sequence. Excuse me. It's located to the N-terminus of the L2, and highly conserved among different human papilloma viruses, or HPVs. So cleavage is not required for virus assembly or release, but essential for infection of new target cells. For example, LOVO and CHO cells lacking furin expression are completely resistant to infection with pseudoviruses of HPV 16, which is one of the high-risk HPV types causing cervical cancer. So HPV 16, one of the high-risk viruses that can cause cervical cancer, and I believe by extension rectal cancer as well. So these LOVO and CHO, these are just different cell lines, and if they have, basically these are what I assume are immortalized cells. I haven't looked into these ones particularly, but these are cells that you can have in a petri dish, or you can have in glass in vitro, and you can test to see if you can run tests on these cells that you couldn't do on people. This is a way that you can learn about the physiology. And so they, when you take these cells and they have removed the ability for these cells to produce furin, these cells then become resistant to the HPV-16 or this particular strain of HPV. So what they're saying is for HPV-16, which is the one that causes cervical cancer and what I assume rectal cancer as well, these cells are immune because they don't express furin. So furin has at least it is necessary for the replication of HPV. So continuing, this illustrates that also non-enveloped viruses have evolved the ability to exploit furin or related PCSKs for their own purposes. So let's make sure I'm not, just, a proteoglycan. I don't know where it was up here. Oh yeah, heparan sulfate proteoglycan. So a proteoglycan is a protein that is bound to a large carbohydrate, and in this case, heparan sulfate. And in contrast to glycoproteins, the protein is larger than the carbohydrate in a glycoprotein. But in a proteoglycan, the carbohydrate is much bigger. So, but it has a protein attached to it. So that's the difference between a proteoglycan and a glycoprotein. And non-enveloped means there's no envelope, and enveloped viruses are usually use the cell membrane of their victim cells to make the envelope. So here, for instance, this is an enveloped virus, HIV. It's using the cell membrane of the cell to make this envelope. Whereas with HPV, this is just pure protein. This is a pure protein capsid. But even these pure protein viruses without an envelope, even these are utilizing furin to get into the cell, which is, their thesis. So hepatitis B, and this is the last virus. So good, we're making good time. Not really. So hepatitis B virus. Another non-enveloped protein that is cleaved by furin is the external core antigen HB-Ag of hepatitis B virus, HBV. So cleaved HB-Ag is secreted from infected cells and exerts immunosuppressive effects. It has been suggested to act as a T-cell tolerogen that prevents killing of infected hepatocytes by cytotoxic T lymphocytes. So stepping back, an antigen is a piece of a pathogen that you can produce or generate antibodies against. So that's just what an antigen is. So, and with viruses, very frequently, the antigen of the virus is the surface proteins of the virus. And so for the case of HBV, this HBeAg or HBeAg is the external core antigen or the external core proteins that your body can produce antibodies against. But apparently if they're cleaved, they can cause immunosuppressive effects. So tolerogenic means that it suppresses the immune system, so it helps you it causes you to tolerate something. It generates tolerance, if that's a good way to describe it. And hepatocytes just mean mature cells of the liver. So hepato means liver and cytes mean a mature cell. So hepatocytes are just liver cells. So continuing on, in contrast, uncleaved HBeAg may have the opposite effect as it is transported to the plasma membrane where it can trigger antiviral immune responses. So if the HBeAg is not cleaved, then it is easy for your immune system to recognize it and attack it and make antibodies against it. Thus, furin-mediated cleavage of HBeAg may affect the outcome of infection. Intriguingly, the Han Chinese frequently harbor a single nucleotide polymorphism in the P1 promoter of the FUR or the furin gene that is associated with increased risk of developing persistent HBV infections with detectable amounts of HBeAg in the serum. Let's break this down. So, a polymorphism is a variant of a gene that is in more than 1% of a population. So within species, there's usually very, very, very little genetic variety within species. We're talking over 99.99% of your genes in your body are going to be identical to the genes of another human. So there's very little genetic variation, even if there are large physical differences. So there might be very large physical differences between you and another person, but those large differences can come from very small actual numbers of mutations or differences within the genes. So most genes, however, there are sometimes multiple variants of the same gene, and these are called polymorphisms if they are in a large enough percentage of the population. So the Han Chinese, I'm not going to get political, but the majority ethnicity of China is the Han people, and they have a unique promoter which can cause differential furin production. Therefore, a disease that utilizes furin as an important protease might have differing consequences between the Han Chinese and other people. So, to do a recap of this, you have your DNA, and the DNA gets transcribed into RNA, and then the RNA gets transcribed into protein, and then continuing on. So DNA has these things called promoters, and these promoters are before the gene. So what ends up happening is things will bind to the promoters on the DNA, and that can tell the DNA to initiate and start transcription of that gene. So for the Han Chinese, they have the FUR gene, the FUR gene, which encodes for furin. And the furin has a promoter right before the furin gene. So the Han Chinese have a small change in the promoter. However, this small change in the promoter of the furin gene can have a different effect than the average person. So the Han Chinese frequently harbor, so let's say you're here, you are a Han Chinese person, and you have this unique promoter. If the, how do I phrase that? Well, I think I need to continue on a little bit more before I explain it. So this polymorphism increases the binding efficiency of the hepatic transcription factors. So hepatic transcription factor NFE2, transcription factor, a factor that influences transcription. So going back here, this red is the transcription. So a transcription factor is going to influence the rate of transcription. So since this particular transcription factor can bind much better with the P1 promoter of the furin gene, if this is expressed, it's going to produce more furin. So thereby, this is why they conclude, thereby most likely increasing furin expression. So whether the observed increase in HBV persistence is the result of increased HbAg processing and or other effects of furin remains to be determined, but the Han Chinese have higher rates of persistent hepatitis B if they, or I should say the persistent production of hepatitis B in their livers, which could imply that the fact that they have this promoter, which is more sensitive to, which is a lot more sensitive to the NFE2, that might be producing more furin in the liver. And if it's producing more furin in the liver, then presumably that furin has a result or is causing an effect on how the HbAg is being activated by the cleavage. So, continuing on, hepatic transcription factor, NFE2, These proteins can bind with the promoter of DNA to influence the expression of that particular gene. So I went through this before. If someone is expressing high levels of hepatic transcription factor NFE2, then the promoters will be activated in many cells around the body, including the liver cells infected with HBV. The Han Chinese have a promoter that interacts more strongly with the transcription factor on the furin processing gene. So therefore, if a virus is in a cell that is expressing more furin due to the NFE2, the Han Chinese might be influenced more than the typical human. Which we know that this is the case. Whether or not furin is causing it is not completely clear, but it is, we assume, Occam's razor. If you're close enough, it's worth it. It's worth making that connection. And then one question I like to bring up is, do other liver-infecting viruses do this as well? So if we try to step back away from hepatitis B, maybe there are a lot of other viruses that can infect liver cells. And if that is the case, is it going to affect the Han Chinese a little bit more than the average person? Some people, we do know that SARS-CoV-2 does infect some liver cells, but I looked into it once, but we'll see. I'll get another paper and talk about that later if I do end up running across it. Done. Okay. Summary. So I'm not going to go over the rest of the paper or the rest of the review. If you want, you can go to the website that I provided to look at it. But here's a summary. Number one, many pathogens utilize proteases of their host organisms. Number two, mono and dibasic cleavage sites are generally more specialized and in less pathogenic, or I should say they are less pathogenic than polybasic cleavage sites. This is because polybasic cleavage sites can be cleaved by many different types of proteases within a host. And by proxy, not just you as a human host, but a lot of other creatures as well. This strategy allows viruses to infect more cells than usual. This strategy also allows viruses to infect a wider variety of hosts. For example, mosquito-borne pathogens often have the polybasic cleavage site. I will do another video on this because that's important to recognize as well. Polybasic cleavage sites can easily mutate from monobasic sites with polymerase slippage. So there is a mechanism by which that you can make the polybasic sites out of the monobasic sites. And that's something that we saw with SARS-CoV-2. So SARS-CoV-2, unlike SARS and other closely related coronaviruses, has a polybasic cleavage site in the place of a monobasic cleavage site. Therefore, it is more easily transmitted and possibly more easily transferred to a wider variety of species. So this is the summary, and I'm going to go into a little... the next slide is going to be pretty technical, so you can stop here if you want. But here I went to Uniprot, which is a database, and you can look up a bunch of different genes that people have put into this database, and you can align them up together, and you can see differences between them. And here, this is all for the SARS-CoV, or I should say, the spike protein of coronaviruses. So here we have the name, or I should just say, here we have the amino acid sequences of the spike proteins. And these spike proteins are all pretty closely related to each other. And I should say the spike proteins are the proteins that allow the coronavirus to stick to the cell. And these proteins need to be cleaved. So sometimes they have monobasic cleavage sites and sometimes they have polybasic cleavage sites. So up here I have the spike for SARS-CoV-2, and you can see here it has arginine, arginine, alanine, arginine. And this is the polybasic cleavage site. And I have put in 4 closely related bat coronaviruses, and as you can see, the closest related ones, at least on this particular database, do not have the polybasic cleavage site. Oh, I should also mention that I highlighted the basic amino acids in green. So here you have histidine, here you have lysine. Those are also basic amino acids, but arginine is the real kicker for the polybasic amino acid cleavage site. So I wanted to include the spike protein of the pangolin, pangolin coronavirus, except it was not within this particular database, or at least it wasn't easily findable in this database. So I really want to, apparently the spike of pangolin is the most similar to the spike of the SARS-CoV-2 coronavirus. So I really wish that I could have that included, because I do know, I saw a different alignment. The pangolin does not have a polybasic cleavage site. It has a single arginine. So just keep that in mind, that the pangolin version, which is the closest version to SARS-CoV-2, is different in that regard. So continuing on with R9. So when we get to the here, this is the MERS-like coronavirus. And shoot, I should have organized this a little bit more. As you can see, it also has a polybasic cleavage site. So RSVR, arginine, serine, valine, arginine, and then another serine. So this has a polybasic cleavage site, and I accidentally duplicated it there. So you can see in MERS, or MERS-like, which I think is about 30% identical. The spike of MERS is about 30% to 37% of the spike of SARS-CoV-2. This also has the polybasic cleavage site, when the more closely related ones do not have the polybasic cleavage site. So, and then here we have the original SARS right here, and the original SARS, with how dangerous it was, did not have the polybasic cleavage site. So it had to use a more specific protease to enter the cell, and this was the TMP-SS2, I think was the name of it. So, but this is one of the reasons why the original SARS was not nearly as infectious as this current SARS that we have going around right now. So SARS-2 is much more infectious than SARS-1, unfortunately. So, and continuing down to the bovine coronavirus, which is right here. So bovine and giraffe coronaviruses, I put these here because these have very, very polybasic, or very basic polybasic cleavage sites. So they have lysines, they have multiple arginines, they have quite a lot. And this is bovine coronavirus does infect a wide variety of species. So there is at least one human coronavirus that is a direct descendant from a bovine coronavirus within the last few hundred years that causes intestinal problems. So I think it was originally called human enteric coronavirus, but it is now, after genetic studies, we realized, oh, this human coronavirus is actually very closely related to bovine coronavirus. And I might do another video on this later, but the bovine coronavirus, which as you can see is pretty much the giraffe coronavirus, is probably also a direct descendant of the bovine coronavirus. These infect many different large mammal species. So we can see here, with some of our confirmation bias, the polybasic cleavage site seems to be allowing this bovine coronavirus to cross species relatively easily. You know, whatever baseline is, though, we're not sure. So continuing, oh, I also wanted to add two of the human coronaviruses as well that cause the common cold. So here we have OC43, which definitely does have one. It has lysine. I don't remember the sequence, but lysine and two arginines. And so this is definitely, Oh, and then also arginine SR is also going to be a polybasic cleavage site. So this is going to be effective. It's going to be very effective at being cleaved by furin. And but the common cold's not going away. The common cold is an endemic virus that it's infectious enough that even though a lot of us have immunity to it, it is still spreading, it is still existing in the population, and it's not going away. Same thing goes for the human coronavirus NL63, and I want to include this one because this also uses the ACE2 receptor, but obviously not as dangerous as the SARS-CoV-2 coronavirus that is going around now, but this doesn't have a polybasic cleavage site, but also uses the ACE2 receptor. So I should also say that the OC43 spike, or the OC43 coronavirus, I don't believe uses the ACE2 receptor. So it's not a good comparison with the current SARS virus that is going around. But this NL63 is a good comparison because it also hooks on to the ACE2 receptor of the human cell, and it's not nearly as dangerous. It's not as pathogenic. It stays within the lungs whenever you get infected with it. So here we have an example of a virus using the same receptor. Yes, it's distantly related, but it's using the same receptor, but it's a lot less pathogenic. And you might say this is just, you know, a dibasic site. It's not following the template or the motif of the polybasic cleavage site of furin, at least. So this is not going to be cleaved easily by furin, but it will be cleaved easily by a trypsin-like protease. So I want to include this. So this is the type of analysis that a lot of phylogenists will be looking at to see, does this polybasic cleavage site actually increase pathogenicity? And it turns out in many cases, yes, it does. So if you want, you can go to Uniprot yourself to I should say Uniprot yourself to look at this, excuse me, for yourself if you want to do an alignment study. So discussion, I did a previous video regarding mosquitoes, and this is somewhat of a continuation on that, because with that previous video, I went over the commentary of a professor. I don't know if he's a professor, but I went over the commentary saying that if the SARS-CoV-2 coronavirus were to become mosquito-borne, become a mosquito-borne illness or an insect-borne illness, it would require some very important mutations that would allow it to infect or at least be able to multiply within a mosquito vector. And this is, since there are no known coronaviruses, and we might not just be studying it enough, but there are no known coronaviruses that can multiply effectively in mosquitoes. But we need to worry in that if we let the SARS coronavirus 2 take advantage and multiply to a large number of people, then we have created the opportunity. We've created a lot more opportunities for this to become a mosquito-borne illness. Because the more people who are infected, the more people will end up getting bitten by mosquitoes. And it'll only take one mosquito, I shouldn't say one, but it'll only take a few mosquitoes biting an infected person who has a mutation that allows it to multiply in mosquitoes. It just takes that event to create a mosquito-borne version of the SARS coronavirus 2. So there are a lot of people that say we should just go for herd immunity as quickly as possible. For one, herd immunity is impossible. You cannot get herd immunity without a vaccine. This is why we vaccinate kids, because as old people die and as new people come into this world, the percentage of people who have immunity to a specific disease ends up getting lower and lower. And then once it gets below herd immunity, then you have another spike in infections that happen. So usually when you're hovering around herd immunity, those spikes are not nearly as severe as the epidemic phase of diseases. But with this particular virus, it looks like it's going to be endemic. It looks like it's going to be absolutely impossible to stop everyone from even wanting to get the virus as well. So my concern right now is we need to limit the ability of this virus, which is highly infectious. We need to limit its ability to cross species barriers again. If it can cross again into mice, for instance, then mice will get infected, and then there will be another reservoir host where it then can jump back into humans again. But I'm very concerned about mosquitoes and insects, because if it can make the cross into mosquitoes, mosquitoes don't just bite humans, mosquitoes will bite many different creatures. And since we know that with SARS coronavirus 2, it can exist in the blood, the virus can exist in the blood, that probably means that an infectious dose could be given to a mosquito. And so if people are, say, homeless or in refugee camps, or just live in locations without a good barrier between their living quarters and the outside insect world, we might run into mosquitoes, or this virus being able to mutate into infecting insects like mosquitoes. And that is not something that I want to see, which is why I'm really glad that we've taken as many steps as we have to help stop the spread of this particular virus while we did. Truly, we did so much that I never thought that we could. So, anyways, so I think, so yeah, that's everything that I want to say. So if you want, you can like and subscribe, and you can subscribe to notifications, because I am going to be putting more videos on the subject, going into other reviews and other papers, and help break them down for people. So anyways, thank you for coming in.